This protocol is becoming increasingly important due to the high number of new self-incompatible Japanese plum cultivars with unknown pollination requirements. The main advantage of this technique is that it allows for the determination of the pollination requirements of cultivars by combining two complementary approaches in field and laboratory conditions. This approach can also be applied to determine pollination requirements in other self-incompatible fruit tree species like apricot or sweet cherry. For pollen extraction, collect flower buds at balloon stage according to stage 57 on the BBCH scale. Remove the anthers using a plastic mesh and place them on paper at room temperature for 24 hours until anther dehiscence. Sieve the pollen grains through a fine mesh and conserve them in a 10 mm glass tube with a cap at 4 degrees Celsius until use. When 10 to 20% of the flowers are open, select and label several branches, leaving 1,000 to 1,500 flowers per pollination treatment. On the next day, when more flowers are open, pollinate each flower using a paintbrush with pollen from the same cultivar for self-pollination and pollen from other cultivars as cross-pollination control. Pollinate every day until all flowers are open. Record weekly counts of flowers and developing fruits from anthesis to harvest to characterize the fruit drop pattern and quantify the final fruit set in each pollination treatment. Collect 50 to 100 flowers at balloon stage according to stage 57 on the BBCH scale. Emasculate 30 flowers per treatment in the laboratory for self and cross